Work Package 6 is dedicated to the study of telomeres in capability and well-being. Telomeres are structures that protect the ends of the chromosomes. As the cells divide, the telomeres get shorter and shorter, and the speed of this shortening is an indicator of the levels of stress in the cell. It comes a point when the telomeres are so short that they cannot keep anymore the integrity of the end of the chromosomes, and therefore they are going to trigger a series of events that are going to lead to cell death. So there is a strong connection between telomere shortening and cell aging. The main research questions for us in Work Package 6 are first, to understand the methodological issues regarding the use of telomeres as a biomarker. What are the limitations of us using them as a biomarker of aging, and how can we overcome those limitations? A second research question for us is to find out how genes and other factors that we know affect human aging through life, such as diet or lifestyle, how these factors are going to affect the length of the telomeres that we measure in adults and old people, and also how affect the rate of shortening of the telomeres. And we are going to do this in collaboration with Work Packages 1, 4, and 7. We also want to find out whether telomeres or the change of telomere length with time is going to be a good predictor not just for aging, but also for changes in capability and well-being in a large group of individuals. And we are going to do this in collaboration with Work Packages 1 and 2. And the fourth research question for us is to test whether the kind of predictions that we are going to do with the telomere data can be made more precise by using other data from other markers such as hormones or gene variations. And we are going to do this in collaboration with Work Packages 1, 2, and 7. So in total, so far, we have 6,000 individuals for which we have one measure of telomere length, and for more than 2,000 of them, we have a second measure of telomere length with time. With such an incredible and unprecedented amount of data, what we have been able to find so far is quite new and interesting. For example, we know that individuals of the same age, they will have different values of telomere length because we don't have all the same starting point. And one could expect that individuals, for example, who are young and of the same age, the variety of telomere length that we are going to find is smaller than the variety of values of telomere length that we are going to find in old people, for which a whole range of genes and environmental factors have had a very different effect through time. And in fact, our data seems to suggest that the opposite is true. The older the cohort that we study, the narrower is the distribution of values of telomere length that we find. And it seems that there is a lower threshold that doesn't shift much with age. So there are individuals who have short telomeres, and through time they manage to maintain them really well. And there are other individuals who have long telomeres, and their telomeres will shorten faster with time. And we think that maybe there is some genetic component to this lower threshold, say that Scots wouldn't have the same threshold than Bromis or Geordies. And it's because we've been able to work with such a large amount of individuals from many different areas throughout the UK that we are going to be able to use much more precise predictions on when we use telomeres as biomarkers of aging. So we decided to organize a multi-center study, fully blinded, whereby 10 samples of DNA were going to be sent to 9 different leading labs that we are going to measure telomeres applying 3 different techniques. One of the things that we are going to do next year is to add another group of samples to our collection of telomere length, and is what we call the carphilia cohort, that includes 1,000 individuals for which we have 2 different samples of DNA at 2 different time points. So that's going to increase enormously the power of the kind of predictions that we are going to be able to make in terms of changes of telomere length with time, but also in terms of the comparisons that we establish with other regional groups. Most of the medical interventions in old people increase the risk of making them frail, a bit more than they already are. So geriatricians will welcome predictive markers of biological aging that are going to help them evaluate the risk of precipitating frailty in old people, and is going to guide them through the right therapeutic decisions. There's been a long-standing quest for a sensitive and specific biomarker of biological aging. And when we put together the results from our work package with that from other biomarker studies in parallel cohorts, we will be able to make an informed decision whether or not telomeres are a good predictor of biological aging.